Bismillahirrahmanirrahim. Assalamu alaikum. Prokaryotes. What are prokaryotes? First of all, you must know about prokaryotes. Prokaryotes are those organisms which have prokaryotic cells in their body, and prokaryotes are bacteria and cyanobacteria. Let's study first of all bacteria. The Dutch scientist Antony van Leeuwenhoek first discovered bacteria in 1674 from the drop of water. It means Leeuwenhoek first discovered. What he discovered? He discovered bacteria in which year? 1674. He called them as animalcules. It means organisms. Animalcules means organisms which we only see by using microscope. Microscopic organisms are termed as animalcules. Ehrenberg introduced the name bacterium in 1882. As you know that earlier living organisms are grouped into five kingdom, but now the status of classification has been changed. This work has been conducted by Carl Woese in 1990, and his colleagues proposed the term domain system of classification. It means now domain system of classification instead of five kingdom classification. This classification, what is the basis of this classification? This classification is based upon the difference of ribosomes, as you know that about ribosomes, and the cell membrane, which is the outermost layer in the case of animal cell. Okay, ribosomes, and this classification on the basis of ribosomes and cell membrane. Now, according to this classification, domains Archaea, bacteria, eukarya. In eukarya includes protista, mycota, it means fungi, plantae and animalia. Let's study features of Archaea. Composition of cell wall. The cell wall of Archaea do not contain peptidoglycan. Peptidoglycan, it is present. This is present in the case of bacterial cell wall, but in the case of Archaea, there is an absence of what? Peptidoglycan in their cell wall. In some Archaea, the cell wall is composed of polysaccharide. Polysaccharide means in some, when we study some Archaea, we have found polysaccharides in their cell wall is pure protein, and in other cell wall is present, cell wall is composed of pure protein. In few there is no cell wall, means absence of cell wall. Mean in some Archaea there is no cell wall, absence of cell wall. The cell membrane of Archaea contain unusual lipids that enable them to function at high temperature. It means these bacteria, these Archaea, these are survive at high temperature because of unusual lipids that is present in it. Lipid of Archaea contain glycerol linked to branch chain of hydrocarbons. Means glycerol, the molecule of glycerol is branched chain linked with the help of branch chain of hydrocarbon. Let's study methanogenesis. Methanogenesis is ability to form what? Methane. Methane is a type of metabolism that is performed by some Archaea. Mode of nutrition, how they obtain food. Archaea use three sources of energy to prepare their food by using sunlight. The first one is using sunlight by the process of photosynthesis. The second using inorganic compounds, and the third using organic compounds. Ribosomal RNA sequence. Now ribosomes. There is a difference on the basis of ribosomes. The most basic difference between Archaea and eubacteria in their nucleic acid. Let's study what is the difference in their nucleic acid. Let's study. Near nucleotide number 910 out of 1500 and one type of rRNA, ribosomal RNA, researchers have found the difference. Archaea, this is a sequence on Archaea: adenine, 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 cytosine, uracil, uracil, adenine, adenine, adenine. In the case of eubacteria, adenine, 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 cytosine, uracil, cytosine, then adenine, adenine, adenine. This is a difference. In the case of Archaea, we have found cytosine, uracil, uracil, and in the case of eubacteria, we have found cytosine, uracil, cytosine.